Assalamualaikum warahmatullahi wabarakatuh. My name is Syazwani Binti Zahidan. My metric number is 1170354. Today I am going to present my proposal regarding evaluation of marine Diabromyces hansenii strain as yeast antagonist against Fusarium, oxysporum and Candida albicans. This is the outline of my presentation. For the introduction, as we all know, agriculture industry is very important to our economy. But the major problem of this industry is to maintain the quality of post-harvest during transportation and storage. This is because it is exposed to diseases, so farmers and producers tend to use chemical substances. Therefore, the Diabromyces hansenii strain is suggested to be the biocontrol agents. Research problem. The first research problem is agriculture industry where fungal contamination will degrade the quality of post-harvest products. Second problem is pest control where chemical substances used have the long-term side effect on consumer health. For the research purpose, the first is to study the antagonistic activity of killer toxin produced against pathogenic fungi and to determine the production of cell wall lytic enzyme. First objective is to screen and evaluate the antagonistic activity of killer toxin produced against pathogenic fungi, which is Fusarium, oxysporum and Candida albicans. Second objective is to determine the production of cell wall lytic enzymes such as chitinase, protease and cellulase. Literature review. Agriculture industry is an industry of cultivation of animal, plant and fungi for human use. In this industry, pest control are used, which are physical control, the use of tillage, open field burning, and also heat treatment. Biological control is the use of natural sources, and chemical control is the use of pesticides. Plant pathogenic fungi are species that obtain nutrients from other plants and give bad effect towards plant health. Can be classified into three, which are biotrophic, necrotrophic, and hemibiotrophic. For example, Fusarium, oxysporum, and Candida species. Antagonistic activity is the action of suppressing or inhibiting the growth of pathogens. It is reported that the Diabromyces hansenii have the ability to suppress and inhibit the growth of human pathogens and plant pathogens. For the production of hydrolytic enzyme and volatile compound, it is identified that 15 different volatile compounds are produced by Diabromyces hansenii, and it is also reported that it has the ability to synthesize beta-glucosidase. Next, material and methods. First, cultivation and growth of marine Diabromyces hansenii strain. The yeast will be grown on yeast mold agar and incubate at 25 degrees Celsius for 72 hours. Second, cultivation and growth of pathogenic fungi. Fusarium, oxysporum and Candida albicans will be grown on acidified potato dextrose agar. And Fusarium oxysporum will be incubated at 21 to 28 degrees Celsius for 6 days, and Candida albicans will be incubated at 37 degrees Celsius for 2 days. Third, the preliminary screening of antagonistic activity of marine Diabromyces hansenii strain using agar plate inhibition assay. So, the Fusarium, oxysporum and Candida albicans will be inoculated on one side of the yeast mold agar, while the Diabromyces hansenii strain will be inoculated on the other side. So, the plate will be incubated for 7 days at 25 degrees Celsius. And after the incubation time, the inhibition of radial growth will be measured and calculated using the formula. Fourth, the production of killer toxin by double filtration. The Diabromyces hansenii strain will be cultured aseptically into the Allen-Meyer flask, where the flask will be containing 100 ml of yeast mold broth of pH 4.5. And then the culture will be incubated aerobically with gentle shaking at 130 rotation per minute for 72 hours at 25 degrees Celsius. Next, the biomass will be separated from supernatant by centrifuge at 5000 G for 10 minutes at 4 degrees Celsius. Then, the toxin sample will be first filtered using the millipore filters, and then the toxin sample will be further filtered using ultrafiltration. Fifth, evaluation of antifungal activity of killer toxin extracted from the Diabromyces hansenii strain by this diffusion method. So, the pathogenic fungi will be swabbed on the Mueller-Heaton agar and dried aseptically for 15 minutes in the laminar flow cabinet at ambient temperature. Next, 25 microliter of killer toxin will be pipetted onto the sterile antibiotic paper disc. Next, the paper disc will be placed on the plate and incubate at 30 degrees Celsius for 72 hours. And after the incubation time, the diameter of inhibition zone will be measured. 6. Evaluation of antifungal activity by fungal biomass method. First, 10 ml of PDB and 10% or 20% of kiletoxin will be transferred to sterilized universal bottle. Then 0.5 microliter fungal spore will be added and incubated to orbital shaker for 1500 rpm at room temperature for 7 days. The fungal culture will be filtered and dried until constant weight is obtained. Now, the percentage of the inhibition of fungal growth will be calculated using the formula. 7. Production of cell wall lytic enzyme. First, chitinase will be tested on colloidal chitin at pH 4.7. Next, protease will be tested on agar plate containing skim milk powder, tryptone, yeast extract, glucose, and bacterial agar. Next, cellulase will be tested on agar plate containing yeast extract, peptone, carboxyl methylcellulose, sodium salt, and agar. And 3 microliter of yeast culture will be inoculated, and the enzymatic activities will be observed after the incubation at 28 degrees Celsius for 5 days. For the expected outcome, the Biomass hansenii Binti strain and the kiletoxin produced is expected to show the antagonistic activity against Fusarium, oxysporum, and Candida albicans. And it also expected to have the ability to produce the cell wall lytic enzymes such as chitinase, protease, and cellulase. So this is the Gantt chart or the plan for the whole final year project. And these are the main references. These are also the references. That's all from me. Thank you.